inheritance is possible because of chromosomes. These chromosomes come in pairs, one from mum and one from dad, so they're called homologous chromosomes. Each chromosome has genes, which are segments of DNA that carry genetic information for a specific trait, and different versions of the same gene are called alleles. As an example, brown eye colour and blue eye colour are both alleles for the eye colour gene, and each parent offers one allele of a gene. Now, these alleles can be either dominant, often represented with a capital letter, or recessive, represented with the corresponding lowercase letter. The difference being that it only takes one dominant allele for its traits to be expressed, whereas it takes two recessive alleles for its traits to be expressed. Human somatic cells, that is, all of the cells aside from the sperm and the eggs, which are called gametes, have 23 pairs of chromosomes, 22 somatic pairs and one sexual pair, adding up to 46 chromosomes in total. These chromosomes, along with the alleles they carry, segregate during meiosis, which is the process of making new gametes. Gametes only carry half the genetic information of the parent, so 23 chromosomes. Once the male and female gametes merge during fertilization, their alleles combine to form the genotype, or genetic information, of the new organism. For every gene, alleles can combine to give rise to three possible genotypes. There's homozygous dominant, or big A, heterozygous, or big A, little a, and homozygous recessive, or little a. This determines all of a person's features, or phenotype, such as eye colour, hair colour, or even whether or not they're colourblind. Now, independent assortment means that no matter which alleles an organism inherits for one gene that codes for a trait like eye colour, it won't affect the alleles it inherits for another gene that codes for a different trait like hair colour. Let's start with a simple example. Let's represent the eye colour gene with the letter A and the hair colour gene with the letter D. Now, the dominant allele for eye colour, big A, stands for brown eyes, and the recessive allele, little a, stands for blue eyes. On the other side, the dominant allele for hair colour, big D, stands for dark hair, while the recessive allele, little d, stands for blonde hair. So let's say we have a person with a heterozygous genotype, big A, little a, for eye colour, and a heterozygous genotype, big D, little d, for hair colour. The person would have the dominant allele features, so brown eyes and dark hair, even though they still carry a recessive allele for blonde hair and blue eyes. Now, at a molecular level, we know that the eye colour gene is physically located on a pair of homologous chromosomes, and in this case, let's say that the chromosome from mum carries the dominant allele, big A, and the chromosome from dad carries the recessive allele, little a. Similarly, let's say that the hair colour gene is actually physically located on another pair of homologous chromosomes. And let's say that the chromosome from mum carries the dominant allele, big D, and the chromosome from dad carries the recessive allele, little d. Now, in meiosis, different pairs of homologous chromosomes independently separate into different gametes. In other words, how one pair of homologous chromosomes splits into daughter cells does not affect how another pair of homologous chromosomes decide to split into those same daughter cells. As a result, a person that has a heterozygous genotype for both hair colour and eye colour can produce 4 different types of gametes. One that carries 2 chromosomes from the mother, and so has the dominant alleles big A and big D. One that carries the 2 chromosomes from the father, and so has the recessive alleles little a and little d. One that carries the first chromosome from the mother and the second from the father, and so has the alleles dominant A and recessive D. And one that carries the first chromosome from the father and the second from the mother, and so has the alleles recessive A, dominant D. So far so good, right? But let's bring 2 more genes along. One that determines skin colour. Let's represent that with the letter B. So with the dominant allele for skin colour, big B stands for dark skin, and the recessive allele, little b, stands for white skin. And another one that determines the type of earwax someone has. Let's represent that with the letter C. So the dominant allele, big C, stands for wet earwax, while the recessive allele, little c, stands for dry earwax. Now let's say that these genes are physically located on the same chromosome as eye colour gene, A, and that the skin colour gene, B, is really close to A, while the earwax gene, C, is far away from A, like at the opposite end of the chromosome. Now, since the 3 genes are on the same chromosome, you might think that they'll all be inherited together, but it turns out that this isn't always the case. The reason is that a funny thing happens in meiosis called crossing over, which is where the homologous chromosomes exchange equivalent parts of themselves which carry the same types of genes, and then after they've swapped, the two chromosomes part ways. For example, let's say that the chromosome that comes from mum carries all the dominant alleles for these genes, so big A, big B and big C, and the chromosome that comes from dad carries all the recessive alleles, so little a, little b and little c. Now, if crossing over occurs between the two chromosomes at the ends of the two chromosomes, then the dominant allele big C that came from mum would end up on the chromosome that came from dad and vice versa. So the chromosome that came from mum would end up carrying the original dominant alleles, big A and big B for eye colour and skin colour, and a brand new recessive allele, little c, for earwax type. Whilst the chromosome that came from dad would end up carrying the original recessive alleles, little a and little b for eye colour and skin colour, and the dominant allele, big C, for earwax type. So this is how, following crossing over, two genes initially located on the same chromosome can end up either as part of the same gamete, like genes A and B, or they can end up in different gametes, like A and C or B and C. Now, crossing over can happen really at any point along the chromosome. So let's say it occurred between gene A and gene B. In that scenario, gene B and gene C would swap over together to the other chromosome, while only gene A would remain on the original chromosome. Now, although this can happen, it's pretty uncommon. And that's because crossing over has an equal chance of occurring at any point along the chromosome. And since there's much more of the chromosome between B and C than there is between A and B, that's the most likely place for crossing over to occur. Another way to think about it is to assign every possible spot where crossing over could occur a number. And for simplicity, let's say that there are 100 potential spots evenly distributed along the chromosome. And let's say that A is a gene near number 5, and B is a gene near number 10, and C is a gene near number 60. Then, if we were to select a number at random where the crossing over would occur, then there's a really high chance that we'd pick a number between 10 and 60, a 50% chance, and a really low chance that we'd pick a number between 5 and 10, only 5% chance. So the bottom line is that the chance that two genes are inherited together really depends on the distance separating them. This phenomenon is called genetic linkage, and this is a common exception to the law of independent assortment. Now, any two genes on completely different chromosomes always have a 50% chance to go through crossing over during meiosis and show up in the same gametes. The same is true for genes that are really far away from one another on the same chromosome, like genes A and C, for example. This is because there might be more than one crossing over event during meiosis. And an odd number of crossing overs would separate them, while an even number would bring them back onto the same chromosome. So, in that sense, ending up on the same gamete or not really just depends on whether there are an odd or even number of crossing over events, which results in a 50% chance. So, for two genes to be linked, the chance that they end up on different gametes has to be less than 50%. Now, of course, even linked genes that are close together might have a small chance for crossing over, if there's a cut right in between them. But of course, that would be uncommon, and that's why the chance that they end up in different gametes falls below 50%. And of course, the closer they are physically together, the lower the chance they would be separated. When it comes to terminology, when linked genes are inherited together, the resulting gametes are called parental gametes, since they carry the same alleles as the original chromosomes. But when crossing over gets between these linked genes, the resulting gametes are called recombinant gametes, because even though the genes were linked, crossing over separated them. For example, if genes A and B gave rise to recombinant gametes in 45% of cases, this would mean that they are only slightly linked, so they're close to one another, but not so much. However, if this happened in 5% of cases, then that means they're really close together, so just really few times crossing over managed to separate them. With genes A and C, on the other hand, they would separate 50% of the time based on the number of crossing over events, so they're not genetically linked. Okay, so as a quick recap, the law of independent assortment states that no matter what alleles an organism inherits for one specific gene, it won't affect the alleles it inherits for another gene that codes for a different trait. In nature, however, there are cases where genes that are close to each other on a chromosome are sometimes inherited together. This is called genetic linkage, and it's an exception to Mendel's second law.